Well, thank you very much to the organizers for giving me the opportunity to make this presentation, and to Camilla for the very nice introduction. So what I'm going to talk about is something we've been interested in for about almost 15 years. But this is the impact on mosquito-borne diseases in coastal areas, where we have, as you know, a problem with increasing salinization due to rising sea levels. We see that already in very areas of the coast where groundwater is becoming more saline. But we also have, with climate change, temperature, humidity, and rainfall, all of which have been reviewed by several authors in detail, and you could look those up. But I'm going to talk only about the impact of salinization in coastal areas on mosquito-borne diseases. So this is our fundamental research would really impact on policy and public health efforts. So firstly, does it have a pointer? So most mosquito species that are vectors of human diseases typically develop in fresh water, that is, water that contains less than 0.5 gram per liter of salt. That's our definition that we're using here. And it's potable, it's drinkable. However, 5% of the mosquitoes normally oviposit and undergo pre-imaginal development, that is larval and pupal development, in natural brackish waters, which is 0.5 to 30 grams per liter of salt, and saline water as well in some cases. So that is more than 30 grams per liter of salt, which is roughly seawater. Some naturally salinity-tolerant mosquito species are vectors of arboviral and parasitic diseases. They include, for example, Anopheles atroparvus in Western Europe, which was the vector of malaria in Holland and Britain not so long ago, less than 100 years ago, transmitting malaria. Anopheles equisales in the Americas, which still is a transmitter in the Caribbean and South America. Anopheles sundacus in Asia, which is a transmitter of malaria in Southeast Asia. Anopheles dorsalis in North America for the West Nile encephalitis virus, and Culex species which in Asia and Africa transmit Japanese encephalitis. These normally breed in brackish water, but they're found only in coastal areas. So what I'm going to describe is the adaptation of freshwater vectors of diseases, particularly starting off with the arboviral vectors, Aedes aegypti and Aedes albopictus, which have been typically regarded for a century or so as a freshwater vector, the breeding or developing it, laying eggs and developing in fresh water. We don't like to use the word breeding, it's strictly incorrect. They actually lay eggs and undergo pre-imaginal development, pre-adult development. So these are vectors of pretty nasty diseases like dengue, chikungunya, Zika, and yellow fever. And the WHO estimated recently that almost half the population of the world is at risk of dengue, transmitted primarily by these two vectors, particularly Aedes aegypti. And in 2023, there were 6.5 million reported infections with 7,300 deaths worldwide. There's no specific or treatment at the moment for dengue. There is a vaccine of sorts, which is not terribly effective for various reasons. Now, one of the problems we have in terms of policy is the international and national guidelines for the control of dengue and other arboviral diseases only acknowledge the development in fresh water. So this is true of the WHO today, up to date. and also the CDC in the US, where we do have salinity-tolerant Aedes aegypti and other pictures. So what do we do about that? Anyway, so what's this story about adaptation of Aedes aegypti to brackish water? We're looking at the basic biology of the mosquito. So typical brackish water culicine mosquitoes that develop in brackish water, like Anopheles, Culex tarsalis and the Anopheles acrosalis and albuminus, have specialized recta that excrete hypertonic urine. So they excrete salt. They're specialized to excrete the excess salt that in the larval stages. And this is completely absent in Aedes aegypti, which is a freshwater mosquito. Aedes aegypti larvae, the L4 stage, for example, has been shown almost 80 years ago in Cambridge by traditional physiologists, that they can maintain hemolymph osmolarity for a very, very short period, approximately 30 minutes or an hour or so, in a salinity of 30% seawater, that is 10 grams per liter salt. And they do this by increasing the hemolymph amino acid and iron concentration. So it's a kind of osmoregulation, but very short term. They can't survive for much longer than that. So how do they adapt? And that's been the subject of our work, which was carried out with colleagues in Sri Lanka. So Sri Lanka island of India, and this is the Jaffna Peninsula here in the north. Maximum height of the peninsula is only about 10 meters. So it's very prone to increasing sea level salinization. And the water supply is from groundwater aquifers. So this is the Jaffna Peninsula and the Jaffna city, and some sites here which are not relevant to this presentation. So we first observed that Anopheles culifaces, which is the major vector of in South Asia of malaria, and considered to be a freshwater species, to develop in coastal brackish water habitats in Sri Lanka in 2010. So that was a very interesting phenomenon. Naturally, it was a swampy area of a small lagoon. We then deliberately searched for and discovered Aedes aegypti and albopictus, also obviously regarded as freshwater mosquitoes, developing in coastal brackish water in Jaffna. And we also showed that they also developed in Brunei, which is in the island of Borneo, in coastal areas. Now, since then, the development of the two Aedes species has been documented in coastal brackish water in countries as far apart as Brazil, India, Indonesia, and the US. So this is a widespread phenomenon. But the WHO and US CDC guidelines for Aedes control, which is followed by most countries in the world and in the Americas, continue to focus only on freshwater habitats. And this compromises arboviral disease management. And we also think that the development of, I'll talk about this in detail towards the end of the presentation, the freshwater malaria vector Anopheles stevensi is undergoing a rapid range expansion, including in the Middle East and to Africa. And we think that is associated with brackish water development ability. So that's also adapted. Typically, this was a freshwater malaria vector seen in India. Okay. Now, the kind of brackish water habitats that we've noticed with Aedes aegypti in the Jaffna Peninsula, the abandoned fishing boats, water collected in these fishing boats, abandoned wells, sometimes used for washing fishing nets, and coastal debris, plastic and ceramic ware. And as you know very well, this is a huge problem in many parts of the world, accumulation of environmental debris, partly through poor waste disposal, but also via the sea. So we have an increasing problem, if you like, of collection of brackish water in these debris, which would be a breeding site for mosquitoes that are adapting to salinity. Now, Sri Lanka, the whole island records about 100,000 cases per year. And the Jaffna Peninsula, which is quite small, about 1,000 kilometers square, has a population density which is pretty high of 700 persons per square kilometer, mean elevation of 5 meters and the maximum about 10 meters. So you can imagine what would happen if sea level rises, as is predicted with the collapse of the Antarctic ice blocks. So the Jaffna Peninsula can get 40,000 cases of dengue per in some epidemic peaks. That's quite high. And we also have shown, and the references are given here, which is at the end of the presentation, that you can look up for details. Aedes aegypti larval density in collections is actually most closely related to dengue incidence in the Jaffna city. Here are some interesting and important properties. The brackish water developing Aedes aegypti and Aedes albopictus are infectable with dengue virus by blood feeding, so they can serve as vectors. And these females that are infected, are able to transmit the dengue virus via eggs to the larvae that hatch from them. So we have vertical or transovial transmission, which can continue over several generations. So if these brackish water habitats are not targeted in vector control, like spraying larvicides and so on, they are a potent dengue reservoir, which circulate, which are present during the dry season, and during the rains, when you have peak dengue, they're able to initiate epidemics or large-scale transmission of dengue. The adaptation to brackish water is likely due to the greater availability of brackish water habitats, and also the greater selective pressure in freshwater habitats where you have the arboviral, where you have the mosquito control measures being applied. So there is a selection procedure here. The genetic and physiological mechanisms that allow freshwater aegypti and Anopheles cutis to ovipost and develop into adults in brackish water has not been previously investigated. So this is stable adaptation. What we showed, and the details can be seen in the references in that particular publication, that the Aegypti and albopictus different larval stages have greater salinity tolerance in the Jaffna Peninsula than in the mainland Sri Lanka. And this is actually from a coastal area of mainland Sri Lanka. So essentially, there has been some adaptation already in that small peninsula. What are the biochemical differences? We looked at transcriptomic and proteomic studies. And we showed that transcriptomics studies showed that there is a differential transcription of messenger RNA for cuticle proteins, particularly the RR2 family in brackish water larvae. And this is in laboratory colonies of the brackish water derived mosquitoes. And for enzymes concerned with very long fatty acid synthesis, which is an important component of waterproofing the epicuticle. Okay, so that fits in rather nicely, and chitin synthesis, because we show that the cuticle is remodeled to some extent, it's thicker, and also for chitin binding proteins. There are some changes in signaling in regulatory proteins, which are interesting. The proteomic data, looking at proteins in the cuticle, essentially confirmed or is consistent with the transcriptomic data. And just to show you here the larval cuticle, and we find that in brackish water they are thicker. The procuticle, we can't see the epicuticle by EM here, transmission EM. So they are thicker. details are in the publication. We also find surface changes in the larval cuticles. So in brackish water, they're much more corrugated. And we can show this by atomic force microscopy and scanning electron microscopy as well. Okay, so that is consistent. Interestingly, and this is also very relevant, that the brackish water larvae, L3 and L4 stages, are more resistant to the larvicide Temifos. So Temifos is the most widely used larvicide that people apply to water containers to get rid of the larvae. So they are more significantly resistant, presumably because of the thicker cuticle. We haven't looked at the penetration studies of Temifos. And what is even very consistent and rather nice is when we reverse the salinity, what you call the reversal colonies, the Temifos susceptibility tends to change towards that of the altered salinity. So this is a partially reversible, if not wholly reversible in some cases. So that's an indication that the resistance is due to the salinity in which the larvae are reared. Okay. More recently, and this was published a few months ago, cuticle changes induced in larvae for adapting to brackish water are carried through to the adult mosquitoes as well. And what do we see here? For example, in this case, we looked at laboratory colonies as well as field collected mosquitoes from brackish water habitats. We find that the cuticle, the whole procuticle, is thicker in brackish water adults. And this is looking at the legs, the T1 tarsomeres. And this is the part of the mosquito that actually, adult mosquito, that contacts treated bed nets, for example. So that's where the insecticide, the adulticides, like pyrethroids, would act. And we also showed that when we change the salinity, when we reverse the salinity, you have the expected changes, that the brackish water cuticle becomes thinner and the freshwater cuticle becomes thicker. There's quite a lot of selection going on. I won't go into the details of it. You can look up here. And that with intended generations. So clearly the cuticle thickness is associated with the development of larvae in brackish water. And this is for the adults, cuticle thickness in adults as well. And this is all, the cuticle thickness changes also occurs in the abdomen of the adults. So if you, when you spray pyrethroid insecticides or as a space spray, the abdomen has the largest surface. So that's where the insecticide would act when you use it as a spray rather than as on a treated bed net. Again, we have reversal, that the cuticle thickness does change even in the abdomen with salinity. And here's the other important factor, that these changes in cuticle are associated with changes in sensitivity to pyrethroids, organophosphate, insecticides. There are a variety of pyrethroids and so on. And they actually move the mosquitoes that develop in brackish water to resistant category, whereas in freshwater they are sort of either susceptible or possibly resistant. So that's a significant change. So in other words, if brackish water adults developing from brackish water larvae in coastal areas are more sensitive to pyrethroid, less sensitive to pyrethroid insecticide, they're more resistant. You can imagine the implications there. There are no difference we find between brackish water and freshwater adult mosquitoes. There is a fair amount of attrition when you change from brackish water to freshwater. The attrition lasts one generation or so, where there is an increased depth of larvae, especially the L1 to L2 transition and hatching. But this is much more prolonged when you change from freshwater to brackish water. Okay, details are available in the publication. So our working hypothesis is that structural changes in the larval cuticle to reduce permeability to water and salt to withstand brackish water are then transferred to the adults. And we think there are epigenetic largely and some genetic changes that may be selected during egg hatching and larval development. So, and basically we think that cuticle changes in brackish water larvae and adults confer resistance to reduced penetration of larvicides and adulticides. Now, moving on to malaria vectors, we saw brackish water development of Anopheles cuticiphates, the typical freshwater vector. And it also developed in coastal wells, domestic wells in the peninsula. We also found Anopheles stevensii, this rapidly expanding, range-expanding species, developing in coastal wells and water tanks that were brackish. And Anopheles stevensii has expanded from India to Sri Lanka in 2017, first detected. I think it's probably true it was not detected previously, and there have been pretty good entomologists there. Stevensii spread to the Arabian Peninsula, then Northeast Africa, Horn of Africa, and then more recently East and West Africa within a couple of years. And we think that container adaptation typically was a vector that in rural India used to develop in natural freshwater after rains. But here container and salinity adaptation were important for a number of reasons. And if you look at the spread, you may be able to see why. The spread from India to Sri Lanka occurred primarily in the coastal areas, and the red dots show where they were detected. And across into the Horn of Africa, also refugees and coastal spread. And then later, of course, we're finding it in Kenya and Nigeria and Ghana, and that's probably through container spread. As you said before, we discussed it, brackish water Aedes aegypti and albopictus are infectable with dengue virus, and they can undergo vertical transmission to the larvae. And they Aedes constitute a neglected and perennial reservoir of dengue virus, which can start off epidemics. And I think we think that a similar process occurs with brackish water Anopheles cuticiphates and Anopheles stevensii, like in dengue, that they are a reservoir of vectors. So what are the impacts that you summarize again? We know the effects of temperature, rainfall, and humidity, how they affect mosquito physiology and the geographical range. But rising sea levels is another factor, increases salinization of coastal groundwater and aquifers, as we see in the Jaffna Peninsula, and expansion of brackish water habitats in coastal areas. We also know that environmental pollution, accumulation of plastic and other containers along coasts do cause a problem. So basically salinity adaptation, increased disease transmission in coastal areas. Conclusions, well, we need to think that other mosquito vectors that are freshwater ones will be affected in similar ways to what we've shown in Aedes. We need to extend vector control to brackish water habitats. We need to change the WHO and US CDC guidelines, and we need to monitor larvicide and adulticide resistance and promote further research in this area. So there are the references which you can look up, and I'd like to thank my colleague, Professor Surendran, and other students in the University of Jaffna and collaborators from a number of European universities. Thank you very much.